Protein synthesis. By the end of this video you should be able to, and this is taken from the GCSE specification, explain how the order of bases in a section of DNA decides the order of amino acids in the protein, and that these fold to produce specifically shaped proteins, such as enzymes. You should be able to describe the stages of protein synthesis, including transcription and translation. You should be able to describe how genetic variants in the non-coding DNA of a gene can affect phenotype by influencing the binding of RNA polymerase and altering the quantity of protein produced. And finally, you should be able to describe how genetic variants in the coding DNA of a gene can affect phenotype by altering the sequence of amino acids and therefore the activity of the protein produced. Proteins are made up of chains of molecules called amino acids. Each protein has a particular number and order of amino acids that makes it unique. The amino acid chain folds up, and the variations in the chain means each will form a different and specific shape. Enzymes are proteins, and it is the folding up that gives each enzyme a specific active site, and so only acts like a catalyst for one reaction. A section of DNA that codes for one particular protein is called a gene. It's the order of bases: A for adenine, T for thymine, C for cytosine, and G for guanine in a gene that represents the order of amino acids in the protein. Each amino acid is given by three bases, called a base triplet, or a codon. In the diagram below we can see two codons, C, T, and G, and A, C, and A. Both of these will code for different amino acids. The amino acids are joined following the order of bases in the gene. Each gene contains a slightly different order of bases, which allows it to code for one particular protein. Some regions of DNA don't code for any amino acids, but these are still important for protein synthesis as they help define how long each gene is. Don't forget that all of an organism's DNA, including the non-coding regions, form that organism's genome. Proteins are made in ribosomes, which are located in the cytoplasm. DNA is found in the nucleus and is too big to leave the nucleus. The code for the protein is needed so that the ribosome can produce the right protein. This is the job of messenger RNA, mRNA. Unlike DNA, it's only a single strand and is shorter. Another difference is that mRNA uses uracil instead of T as a base to pair with A. mRNA transfers the code found in the DNA to the ribosome. RNA polymerase is the enzyme responsible for unzipping the DNA strand and joining together the bases which complement those in the template strand. To start transcription, RNA polymerase binds to a non-coding region in front of a gene. The two DNA strands unzip and the enzyme moves along the coding strand. Once made, the mRNA molecule moves out of the nucleus to join a ribosome. This means that if a DNA strand goes CAG, the mRNA strand would be GUC. Once bound to a ribosome, the next stage, called translation, begins, where the protein is assembled. Amino acids are brought to the ribosome by another molecule called transfer RNA or tRNA. The order in which the amino acids is brought is determined by the codons in the mRNA. tRNA also has an anticodon, which is complementary to the codon, ensuring that the amino acids are in the right order. The amino acids are joined by the ribosome to form a polypeptide, which is a posh term for a protein. This diagram shows the transcription process. This diagram shows the translation process. Note the T shape of the tRNA. Mutations are a change to a organism's DNA base sequence. If it happens in a gene, a variant of the gene is produced. The genetic variant may help code for a different amino acid, which might change the shape of the end protein, and thus its activity will be affected. This could end up changing the characteristics of an organism, which is the phenotype. If a mutation happens in the non-coding region, it can result in the RNA polymerase finding it either more or less difficult to bind to the region. This can affect how much mRNA is transcribed, and therefore how much of the protein is made. And depending on the protein's function, the phenotype might also be affected by how much is made. So by now you should be able to explain how the order of bases in a section of DNA decides the order of amino acids in the protein, and these fold to produce specifically shaped enzymes such as proteins such as enzymes. You should also be able to describe the stages of protein synthesis, including transcription and translation. You should be able to describe how genetic variants in the non-coding DNA of a gene can affect phenotype by influencing the binding of RNA polymerase and altering the quantity of protein produced. And finally, you should be able to describe how genetic variants in the coding DNA of a gene can affect phenotype by altering the sequence of amino acids and therefore the activity of the protein produced. And reminder, this is from this specification. If you've enjoyed the video and learned from the video, please like and subscribe. If you have any topics you would like me to cover, please comment down below.